Hello, everyone. Good afternoon, and thank you for joining this webinar on amazing viruses that shape and escape our immunity by Chris Benedict. My name is Mitchell Cronenberg, and I'm the Chief Scientific Officer at the Jolla Institute for Immunology. And because I've been in leadership here for many years, I can't resist telling you a little bit about our organization before we start the main event. The Jolla Institute for Immunology is an independent, nonprofit biomedical research institution. We're a 501c3, tax-exempt, charitable organization. We study the immune system to treat and prevent diseases, and we are highly ranked in the world, top 5 at one point by the Scientist magazine. So our motto is that the Institute will engage in a world-class biomedical research program with a focus on the immune system. And we will conduct, share, and partner such that the results of our discovery program will make outsized contributions to the betterment of human health. And by outsized, I mean we are not very large, but we're impactful, as I'll tell you a little more about. In fact, we have 24 faculty, each one leading an independent laboratory, about 500 total staff, including 200 trainees, high school, undergraduate, graduate students mostly, and postdoctoral fellows, in fact, all of them learning to be world-class scientists. And we come from 35 countries, 27 different native languages. People from all over the world come to work at the La Jolla Institute for Immunology. Why immunology? Most people now know about the role of immunology in infectious diseases and vaccines. But also there are many autoimmune diseases. The immune system can be trained to fight cancer. And the immune system contributes to chronic inflammatory diseases like cardiovascular disease and neurodegenerative diseases. There's one way I'd like you to think about it. If you think of psoriasis, a skin disease, arthritis, a joint disease, ulcerative colitis, and intestinal disease, different end organs. But for each of them, the most effective treatment now, and it's not a cure, is to block an immune molecule called TNF. And you'll hear more about the TNF family later. And so the point I want to make is that all of those diseases and many others have an underlying immune cause. Even though if you have that disease, you might think of the end organ. I have a skin disease, but in fact, or I have a joint disease, but in fact, arthritis has an underlying immune component. What fuels our work? Well, of course, dollars, predominantly federal grants and contracts, other kinds of grants, non-federal, and philanthropic contributions from people who support our work and who are interested in it. So in fact, we are publicly supported, and that's why it's so important to communicate with the public. We also have some licensing income and affiliates, including corporate and the University of California, who help to support us. How about our impact? How can we measure it? Well, 8 of our scientists last year were among the top 1% highly cited. That means that other scientists are reading our work and pointing to it as findings that stimulate their own work. And some of our findings lead to intellectual property, including licensing agreements, commercial sponsored research, patents, and drugs and technologies in clinical trials. The furthest along, probably just about to enter phase 3. Another way to measure our impact is the extent to which we are in the media. Our scientists are interviewed and cited continuously by the leading outlets, particularly during the COVID-19 pandemic, which in fact is not in the past yet. Okay, so our talk today will be about amazing viruses that escape our immunity by Chris Benedict, a member of our faculty. I've known Chris for 25 years, or since he came to the Institute as a postdoctoral fellow, and we've never let him go. He had the idea to study how viruses interact with the immune system over the longer term. and he's a wonderful colleague who many of us collaborate with. And I'll stop sharing and I will hand it over to Chris now. Chris, go ahead, please.
Thank you very much, Mitch. Yeah, it's been a pleasure to be at the Institute for this many years, and then obviously with colleagues like you and our other colleagues at the Institute, it's been an amazing experience. So amazing viruses that can escape our immune system. So when we get infected by viruses, typically like, for instance, SARS, which causes COVID, or influenza, these viruses are ultimately cleared by our immune system completely. But there's other viruses, like the ones that I'm interested in, particularly, that basically are not cleared, but can kind of escape through and establish what we call latency, or this persistent infection. And so that's sort of shown in this diagram here. There's two walls to our immune system. There's the innate immune system that functions in the first couple of days, that sort of dampens down the virus infection, but can't control it completely. And then there's a second wall, which is made up by what we call the adaptive immune system. And the adaptive immune system is composed of antibodies, white blood cells, other things that many of you have likely heard of. Now, in the case of influenza, for example, we can block it very well at this first stage. And at the second stage, we can clear it completely. But in the case of the viruses that I'm interested in, the herpes virus family, which includes chickenpox, Epstein-Barr virus, which causes mono, that many of us have had when we were in high school, and another virus called CYTOMEGALOVIRUS, or CMV, which my lab works a lot on. Basically, those viruses are able to sort of escape through this narrow trap door and establish this lifelong infection. And a great example of that is for those of us who are old enough to have not been vaccinated for chickenpox. We got chickenpox when we were kids, and now we have to worry about getting shingles when we're older. And yes, there are good vaccines that are developed for shingles now, but still, that virus that we got when we were kids with chickenpox remains with us for 50 years. So this is a very interesting dynamic between our immune system and viruses that aren't able to be cleared. So this is a two-party battle, what I like to think about it. Our immune system is fighting the virus is fighting our immune system. So what is it about the viruses that I study that make the immune system unable to clear it completely? So that's a very interesting question. It targets specific arms of our immune system in order to accomplish that. So CMV, or cytomegalovirus, is one of these viruses. 50% of you who are sitting on this webinar right now, if you're in San Diego, are infected with CMV and have probably never even heard of it. Unless you're at risk for immune disease if your immune system fails. So 10% of your white blood cells are specific for this virus, 10%. So there's 1 million cells per every milliliter of blood in your body, and 10% of those are dedicated towards CMV if you're in the half of us that are infected. So it's the number one most known non-heritable cause of immune system flexing, for lack of a better word, right? So in other words, the immune system is always shaping itself one way or the other. And this virus has a particularly big impact on our immune system. And if your immune system is compromised, so for instance, you get a transplant, or in the case of here, it's the number one infectious cause of congenital birth defects. So in other words, if you're a pregnant mom and haven't been infected with CMV before, you get infected in your first trimester, there's a very large chance that your developing fetus will get infected. And in that case, then there is a very big chance that you're going to get some disease if you're the developing fetus. So the CMV, so in this case, CMV vaccine development is a very high priority for the Institute of Medicine here in our country. So CMV has evolved for probably 100 million years or more with our immune system. And that's why this push and pull exists, why we can't clear the virus, but also why the virus doesn't cause serious disease unless our immune system is compromised. So if we want to think about this in terms of different kinds of persistent viruses that our immune system can't clear, hepatitis, HIV, versus CMV or Epstein-Barr virus. In the case of hepatitis and HIV, they're more of a hammer on our immune system. They sort of keep replicating and basically wear down our immune system over time. But if we're CMV or chickenpox, then what we get is more of a sort of a tickling of our immune system, changing it over time, but not completely wearing it down. And so that's an important point to sort of understand. So last thing that I'll raise before we go to Eduardo in my group to talk about what's going on in the lab, is that there's, in my opinion, very big similarities between the way that cancer impacts our immune system and persistent viruses impact our immune system. Persistent viruses have all these strategies to basically target our immune system, dampen them, and don't allow our immune system to clear them. And in the case of cancer, cancer is very similar in many cases, where cancer impacts our immune system in a way, inhibits it, and now there's all these new drugs that have been developed to basically be able to reinvigorate our immune system to fight against cancer. And so that's the way I look at this sort of push and pull between the immune system and cancer, and the immune system and the persistent virus infections. So it's a real pleasure to introduce Eduardo Lucero in my group. Eduardo was born and raised in Baja, Mexico. He got his master's degree from the University of Ensenada in nanoparticles. He's been a fantastic addition, and I look forward to seeing what he has to say.
Hello, everyone. My name is Eduardo, and first of all, thank you, Chris, for such a warm introduction. I'm working here at the Benedict lab as a lab manager, and right now I'm going to show you a little bit what we do in the tissue culture room. For that, we need a biosafety level 2 area, which will allow us to work with human pathogens that could pose a moderate health hazard. So that's it. Let's get in. All right, so right now we're going for the hood, tissue wounds. So let's just get a little bit of ethanol in our hands. Let's keep it clean all the times. So what I'm going to do here inside the hood, which will protect me against whatever pathogens I'm working with inside. This works by maintaining like a very a laminar flow, airflow around the area. So it'll keep it sterile at all moments, while at the same time protecting me for whatever I'm handling here. So right now we have some cells here. I already added some trypsin, which is a detaching agent for the cells, which they were attached to the bottom of the plate. So I will need to do that. So right now, what I'm going to do is to pass these onto this tube. Let me just start doing that. So very carefully, we're going to remove the lid and we'll grab our cells, give it a little help so they can detach from the bottom. Very carefully, gentle, at the same time, give it a little shake. Then we can grab all of our media containing the cells. We are now going to pass it into these two, very carefully. Let's grab this, put it away, also carefully. And now we're going to take this into the centrifuge. So what we're going to do now is to get the cells to go to the bottom of the tube so we can remove the media and add new fresh media. So this way we are basically washing them and passing on to a new vessel. Always remember to keep your balance here. We don't want any accidents with the centrifuge. So they have to be about the same volume and weight. So just give it a couple minutes. So in the meantime, I will talk to you about what we are going to do with these cells. We use them to reproduce our virus and also to determine how effective our virus can be. For that, we perform plaque assays. This is done by just infecting my cells in these 24 well plates, let the infection happen, and then after a certain point of the infection, the cell membrane of the cells here will get disrupted by the infection, so releasing more virus into the media. That disruption of the membrane will form plaques, and that's what we are going to count here. So you can come a little closer here. Yeah, have a few spots there, maybe around 30 or 40, which means we have a viable virus that will infect our cells, and that we can use it for our further experiments. So now, to explain all of this, looks like our centrifuge has done spinning. So let's just give it a couple seconds. And there we go. Right. And let's just grab our cells now. They should be at the bottom. Then let's remove the media. Be careful not to extract your cells from here. There we go. Yeah, we don't want to remove the cells from here. Now I'm going to grab some media, new fresh media, which we're going to use to resuspend our cells. Then we are careful here. Give it a little shake here, stir, stir. There we go. Now we're going to grab some of this pre-warmed media and pass it onto the new vessel. Okay, let me just open this serological picture. Okay, so let's grab about 9 milliliters of this media. This red liquid, it's culture media, which we use so our cells can receive its proper nutrients and growth factors. Now that we have the vessel prepared, we're now ready to plate our cells here. Now we will grab about 250 microliters. Distribute it evenly. Don't forget to always do it like this. Remember, not swirling. If you swirl, the cells will tend to go into the center, and we want them evenly distributed throughout the area. All right, so that's it. Now let's bring it to the incubator. Let's bring it to the incubator first. Listen here. There we go. Now our cells will be incubated at 37 degrees Celsius, or close, which is our average body temperature. We try to keep this as physiological as possible. And now in about 4 days, we'll be able to use those cells for either infection or for a plaque acid. So that's pretty much it from me. Thanks for listening and tuning in. Let's go back to Chris.
Fantastic, Eduardo. Thank you very much. Obviously, everyone can see what a great asset Eduardo is to the laboratory. So just to finish on maybe a bit more detailed talk about what we do in the laboratory in terms of understanding the actual molecules in the virus that target our immune system. CMV carries about 700 genes. Flu is less than 15. So this is a big virus. It's made up of DNA, like our own chromosomes are. And because there's so many genes in this virus, it provides a very good landscape in order to understand how this virus is able to evade our immune system and establish this lifelong infection. So I'm going to talk about 2 particular genes today in the virus that my lab has been interested in over the last 10 years or more. So one of them is called UL141 and one of them is called UL144, like, terrible names, right? Because there's UL1 through 200. But so interestingly, we haven't developed a better nomenclature than UL141 and UL144. But so looking at this, so Mitch touched on this in his introduction. The TNF receptor, a tumor necrosis factor receptor family, is a very interesting family of which our immune system acts upon, and drugs that target this family have been very effective in combating diseases like, for instance, arthritis. So there's another member of this family. There's about 24 genes in this family. One of them is called HVEM. And this molecule of our immune system binds to both a cytokine called BTLA. Remember, cytokines are those that are secreted by our immune system into the blood, as well as it binds another cytokine called LIGHT. And when BTLA binds to HVEM, it actually inhibits our immune response. Whereas when LIGHT binds to HVEM, it activates it. So human CMV has basically stolen this HVEM molecule from our immune system and modified it so that it only binds to BTLA. So now it's only a molecule that can inhibit our immune response. It's not one that can bind to light and activate our immune response, which again, likely contributes to the ability of this virus to escape our immune detection and escape our immune system. So UL-141 binds to a receptor in our immune system called trail. And trail was originally described as being an important molecule in anti-cancer defense. And there have been many clinical trials testing whether if we can activate this trail pathway in cancer can help us clear cancer. So the trail ligand and the trail receptor in our immune system is very broadly expressed. It's actually induced to higher levels by virus infection, which induces cytokines called interferons. And that name might make sense because interferons actually interfere with virus replication and virus infection. So it's been a little unclear exactly how this trail cytokine system of our immune system might be combating virus infection via combating cancer. And so what we found was that the virus CMV encodes this protein, this gene UL-141, which is shown in orange and in green, to bind to our immune system's receptors, trail. So this interaction, which we were able to visualize at a very atomic level, is a very interesting interaction because typically in our immune system, we have 3 immune molecules in the TNF family, shown in blue here, for instance, trail, which bind to 3 of the trail receptors. But in the case of the virus molecule, now we only have 2. We have 2 here. This was orange and green on the last slide, but this is 1 in yellow and a second in yellow that bind to the trail receptor. And this was very interesting to us because 3 molecules binding to 3 is oftentimes very different from 2 molecules binding to 2. And so that's teaching us new things about the way that our immune system signals. So what we can do is we can actually look in tissues where the virus is secreted, where CMV is secreted. For instance, a very common way that CMV is passed between mom and child is CMV is shed in breast milk. And when breastfeeding, then the baby will be infected with CMV. So we have a red virus shown here, and we have green immune cells. And we can utilize this amazing microscope core here at the Jolla Institute to basically look at the specific patterns of the way that immune cells can interact with virus infected cells. And again, that is something that's very unique to the Jolla Institute. And Zbigniew, who runs our core, is fantastic with that. So just to kind of like finish this story, the immune cells that I showed you back here on this slide, the green immune cells, are called CD4 T cells. And these particular CD4 T cells are critical to eventually control the virus from being secreted from the salivary gland, from the kidney, from the breast milk, etc. So basically, what you see is that there are CMV specific cells that fight the virus. But these cells don't do a great job early on during infection. And one of the reasons is because our immune system basically dampens the ability of the virus to be cleared. Now, we found that there are a particular flavor of CD4 T cells that come up later during infection. And these T cells are absolutely critical for the virus to eventually get controlled, stop being shed, and establish this lifelong infection. So to just finish on a broad note, in my opinion, persistent viruses that target and evade our immune system can teach us a lot about what our different immune mechanisms are, and also maybe even broader, what our immune system can be harnessed to do to fight persistent virus infection and cancer. And maybe we'll go to Q&A now.
Yes, yes, we will. Thank you, Chris. That was really outstanding. It's interesting how these viruses have evolved to live with us over the long period. So the first question is, there are millions of people whose lives are made difficult by these infections. Maybe they're immune suppressed. And what is the hope for another medication or cure even, besides acyclovir, valacyclovir, be it a small molecule, be it a biologic, be it a real vaccine? How do you see the landscape in terms of treatment? And related to that, do people develop, or do the viruses develop resistance to acyclovir?
Yeah, firstly, that's a great question from the panel. And yes, viruses do develop resistance to these small molecule drugs. And so drugs like acyclovir or ganciclovir, that some of you may have heard of on the video today, those drugs basically are incorporated into the virus DNA when it replicates and stop it from being able to replicate. But the reason that they're specific for that is that they're able to inhibit the viral DNA polymerase, which is the same enzyme that replicates our own chromosomes when our cells divide. Okay, so basically what happens is that the virus mutates its own DNA replication enzyme and doesn't allow those drugs to work any longer. And so there's a lot of work being done currently to try to find new drugs that the virus cannot sort of circumvent, just like Mitch said.
Yeah. What about vaccines? What's your analysis of the vaccine frontier, or perhaps even using biologics? And biologics means bigger protein-sized drugs rather than tiny molecules that can integrate into another protein and prevent it from replicating DNA, as the acyclovir example.
Yeah, I mean,
how do you see the vaccine landscape?
Yeah, so for instance, the vaccine landscape, one of the most effective vaccines in the last 20 years has been the vaccine against shingles. And shingles, of course, is when the virus reactivates, when you got chickenpox, when you're a kid, 50 years later, it reactivates when we're older and can cause serious uncomfortable symptoms. I actually had shingles a few years ago. So in that case, there's a vaccine that's essentially called Shingrix that's actually 95% effective. So a vaccine approach, in my opinion, is a great approach. For CMV, for herpes, simplex virus, for Epstein-Barr virus, there's all active vaccine approaches that are ongoing at various pharmaceutical companies, because that, in my opinion, is probably the best option.
Yeah. How does the Shingrix vaccine protect? Is it stimulating T cell immunity or antibodies or something else? And does it wipe out the virus?
Yeah, so it doesn't wipe. There's no evidence right now that it actually wipes out the virus. It stimulates both your antibody response and your T cell response. It's one particular protein from the virus that you boost your immunity to. It's called glycoprotein E for the people who are interested in that. So basically, it's a way of boosting your immunity, because probably when you got chickenpox 50 years ago, your immunity has not maintained its level over that time. And now we give someone when they're 50 a vaccine, and it boosts up again to the level where we don't get reactivation of the virus and we don't get shingles.
Great. Yeah. I have two questions that are kind of interrelated. And one is, if you are infected, is there any way to reduce viral load? And maybe you could just touch on transplantation and immune deficiency and what sometimes happens in those cases.
Yeah, so one of the biggest problems before drugs like acyclovir and ganciclovir were developed was that if you got an organ transplant, your immune system has to be repressed in order to accept that organ. And then if you were infected, for instance, with CMV would reemerge and cause pneumonia-like symptoms in the lung, which was a serious problem. Or for instance, if you're an HIV patient, CMV would reemerge and actually cause people to become blind because it would replicate in the retina of our eyes. And so in my opinion, these drugs are still able to dampen down these issues. HIV, for instance, triple drug therapy has gotten to the point where we don't wear our immune system down enough to have active cytomegalovirus infections in our eyes anymore. But I think most likely there's still a lot of yards to be gained in terms of vaccine approaches and other drug approaches.
What about another member of the family, the Epstein-Barr virus, and its relationship to multiple sclerosis? What can you tell us about that? Some people have heard about this, others probably have not.
Yeah, there was a very sort of seminal paper that was published a little more than a year ago, basically saying that all patients who develop multiple sclerosis are EBV infected. And one of the issues with that, which is sort of common to this family of viruses that I study, is that most of us are infected with many of
Yeah.
these viruses. And so whether this is causative or whether this is basically just sort of coming along for the ride remains a little unclear. So what I would say, in my opinion, so what I would say is that, yes, there is a correlative causation between EBV infection and MS. But whether EBV is actually causing multiple sclerosis or whether it's sort of along for the ride, as I said before, still remains a bit unclear.
I have a question in which I have a personal interest. Turns out I had a younger brother who had a neonatal CMV infection and died from it due to extensive organ damage at the age of 5 years old. It was an enormous tragedy in my family. So the question is, do pregnant women and newborns get screened for CMV? And what can pregnant women, because pregnancy may be associated with a certain level of immune suppression, not at the organ transplant recipient level, but still, there's some immune suppression. So what can pregnant women do to protect themselves?
Yeah, that's a great question, Mitch.
I don't ask easy questions.
Yeah,
not me, but the audience doesn't ask easy questions.
Yeah, it's not an easy one, but so what I would say is, for instance, Minnesota has established recently just a pan screening of all newborn infants for CMV infection, just to see, what the incidence of infection is and what the incidence of disease is. And the fact that individual states, Idaho is another one that's doing that now. So the fact that individual states are actually taking it upon themselves to screen pregnant moms, screen newborn infants for CMV infection is going to allow us to develop a database over the course of the next, 20, 50 years to understand what impact that may have, in various aspects of our health and disease. Your brother, Mitch, that's a fairly rare case, obviously. Tragic. That happens to about 100 to 500 kids a year in the United States. More common is congenital hearing loss in infants. But yeah, if the most at-risk group of women are women who are pregnant with their second kid, who are not CMV infected before, and their first kid is shedding CMV, which is quite common. And unbeknownst to them, they get infected as they're pregnant for the first time. And that's the group that basically is usually targeted for clinical trials. So
it's interesting that back in the 1960s at Cornell, they had to go to Cornell University Medical School to diagnose the disease by immunoelectron microscopy. I remember my parents coming home, and I was a little kid myself. What's immunoelectron? They thought I was smart. What's immunoelectron microscopy? I said, I don't know. When's the Yankee game on? But can you give neonates acyclovir?
Acyclovir is more toxic for neonates than it is for adults. They do give it in cases of high risk infections. But again, because the virus is actually replicating a lot in neonates, typically in that case, the virus has a tendency to mutate away from that drug being
Right,
effective.
right.
Yeah,
Wow.
but yes, they do it in high risk cases.
Great. Thank you, Chris. I think we should wrap it up there. The questions you've done, you and Eduardo have truly done a magnificent job. It's a little counterintuitive to think that if we're healthy, we may be carrying around viruses and we're asymptomatic and everything's fine, but they do present some risk. So in conclusion, I thank you again. I'd like to point out that we're funded predominantly by grants, as I told you, but the philanthropic support is very precious to us because there are expenses in equipment and other areas and supporting young scientists that really are not available from grants. And so I want to emphasize that if you are interested, you can make a donation online, or you can contact our Vice President of Advancement, Kelsey Dale. You can look at the QR code here and use that to make a donation. We have many ways. Visit our website. You have many ways to help us if you have that interest. I really thank everyone for participating today. A recording of the webinar will be available and will be sent out to each of you who is registered in the next couple of days. And I would encourage you, if you're interested, to share it with others who may be interested, or to look at it again yourself to learn some of the material. But I promise there won't be a quiz. That concludes the webinar. Again, thank you for coming to listen to us today. Bye-bye.
Bye-bye, everybody. Thank you very much.